Hello, and welcome to the How Epidemics End project based at the University of Oxford. My name is Erica Charters, and in these videos, I discuss how experts research disease in a variety of ways, as well as their investigations into how epidemics end. Today, I'm here with two scientists who study human evolution and ecology in the way that they study disease. So I have here with me Nils Stenseth, who's professor of ecology and evolution at the University of Oslo, and who's an evolutionary biologist and ecologist. And I also have with me Barbara Bramanti, who's associate professor at the Department of Environmental and Prevention Sciences at the University of Ferrara in Italy, who's a physical anthropologist. So Barbara and Nils, I think people might have a general idea about human evolution and how it relates to ecology and to the environment. But can you explain a little bit more specifically how you, as evolutionary biologists and as physical anthropologists, study disease? So Nils, if we're to start with you, do you work in a lab, in field work, on a computer? What kind of evidence do you use in your research?
I myself mostly work on other people's data and long-term time series, so observations over many years. And when I first got into plague work, it was analyzing data from the old Soviet Union about recording of occurrence of plague in wildlife. We have to remember that the plague caused by Yersinia pestis is a wildlife disease that occasionally spills over to the human population. And as a matter of fact, it spills over to the human population when plague in wildlife ends. That's right. So I'm analyzing these kind of data. What is the structure of the dynamics in space and time? And I do statistical models based on really mathematical population models. So that's what I do for a living.
That's very helpful. And Barbara, I know in some ways you also look at plague, but from a slightly different angle. So can you explain how your research works when you do physical anthropology?
Yes, I'm a physical anthropologist, as you have said. That means that I am interested in human evolution and in terms of population dynamics and in terms of human environment interactions. And since disease are part, especially infectious disease, are part of this interaction between humans and the natural environment, I am also very interested in this kind of infectious disease, and in particular on plague. And my group has provided the first proof that the past plagues were caused by Yersinia pestis, as well as the modern plague. And from this point on, we have started to study more in depth the dynamics of Yersinia pestis and the modalities of spreading of the bacterium in the past.
So this is really interesting because, of course, the two of you work together, and I know you work with many other researchers. So can you explain a little bit more about how then you study plague when you're working together? Nils, you've talked a lot about how you're part of the center that brings together different disciplines at the University of Oslo. Explain a little bit about your research and how it works with Barbara's research. What have been some of your findings?
I can start where we sort of got together. I have believed that plague had reservoirs here in Europe because we have, superficially the right condition. However, we did a paper, not with Barbara, but with other colleagues here, that was published in 2014 or 15, I believe, where we demonstrated that the occurrence of plague in harbor cities in Europe was linked to good plague conditions in Central Asia several years earlier. So then I became convinced that, as a matter of fact, plague must have come into Europe several times. And then Barbara was the obvious partner for linking up here, because she was then studying the genetics of old skeletons, or actually the teeth of old skeleton, and you can explain this much better than myself. And that the work that Barbara did and Barbara and we did together really supports our original idea that plague must have come in several times to Europe. So that's really where we met. We understood pretty much how plague ends in wildlife, but we don't know how the dynamics in the human population is. Her project was really spot on to answering that question.
I think this is a really interesting point, and Barbara, I want you to expand a bit on this, but especially thinking about what kind of sources and data you're looking at, because many of us might know that, for example, historians of plague, we've talked to some of them in this project, look at textual sources, so the records that humans are writing and leaving behind, but you seem to be looking at different types of sources to study alongside. So Barbara, what kind of sources then are you looking at? What are you combining? What kind of data are you using when you're studying plague and trying to understand the course of an epidemic?
Yes, as a molecular anthropologist, I work in the lab and I have a group working in the lab, and we try to isolate DNA, ancient DNA from the teeth, and to reconstruct the genomes of the ancient bacteria and to compare them. And by doing so, we can reconstruct also the phylogenetic tree of the bacteria, and we can better understand the dynamics. But the phylogenetic tree is not so easy to be interpreted, and we need also other kind of data. And this is also the reason, because of this interaction, very fruitful interaction with ecologists and archaeologists. We need a lot of competencies, of course, also of historians, because we have always the historical evidence in the background when we try to interpret the phylogenetic tree.
So, and Nils, you've already mentioned something about plague ending, but plague ending only in certain groups or certain, I guess we could say, niches, certain reservoirs. Can you explain a little bit more about what you mean by plague ending and how you measure the end?
Yeah, it ends continuously in wildlife population. Great gerbils is one of the species being the host of Yersinia pestis, a bacterium, in Central Asia. And when condition over a large area is hot but not too hot, humid but not too humid, then the gerbil population become larger and larger and come above a certain threshold. When it's above a certain threshold, abundance, I mean, how many there are, then it is likely that there might be a plague outbreak in the gerbil. Then, if that stays on for a couple of years, then it's where you can have a plague outbreak. Then, over the same area, if there suddenly becomes very dry condition, so that is bad for the vegetation, bad for the flea, bad for everybody, then the gerbil population crashes. And that is exactly the time when the gerbil population crashes. That's when it spills over to the human population. It is very interesting that I know that the plague pandemic in the wildlife ends when it is dry condition, whereas historians, they say that it starts in the human population when it is dry condition. Of course it is, because that's a spillover.
That's a really fascinating point, that, of course, if you're looking at animal reservoirs, and as you say, gerbil populations and how that relates to environmental conditions of climate and so on, but what ends in the animal reservoir then is when it starts in the human population. So Barbara, you look more at human populations. So how do you see plague ending in those populations?
Well, you need also to understand how it starts. If it is because of contact with the animals of a reservoir, you can have from time to time again new cases, because the contact can also be by chance. But if you have an epidemic, you can see that you need some particular condition, which are hygienic condition mostly, and a control, a prevention. We have studied the third plague pandemic in Europe, and we have clearly seen that it was introduced continuously from outside. And this is because we have no plague reservoir in Europe. So the possibility to have some contact with hill animals is very reduced. But plague was introduced from outside, that it was in a period at the end of the 19th century when people were very aware about importation of a new infectious disease. During the 19th century, it was introduced cholera, for instance, and they have developed a good system to avoid the introduction of plague. And for instance, plague became notifiable and internationally notifiable. That means that all the people were aware about the possibility to import plague. So they have the situation under control and have used a lot of measures to avoid the spread of the contagion. For instance, isolation of cases, information to the public and to the healthcare, sanitation, hygienic measures, and in such a way they have stopped the contagion. And we have had, during the third pandemic, only one large outbreak in Europe, but all the other contagion, or all the other outbreaks were very reduced in their dimensions. And from the beginning of the 1950s, we had a stop of the introduction of plague, also because new measures were introduced, like the Hoover, the wash machine, and everything which could destroy the ectoparasites, which are the vector of the transmission of plague. And yes, we are convinced that in the absence of a reservoir, these measures are enough to stop a pandemic or an epidemic.
It's really fascinating too, and I think a good reminder to many of us, because I'm sure many people watching and listening will associate plague with, say, the Middle Ages and the Black Death. But of course, as you point out, there's a third plague pandemic starting in the 1860s, and that which continues on into various parts of the world in the 20th century. And if I've understood your research correctly, as you both point out, the outbreak depends partly on climate and environmental conditions, which also then affects animal reservoirs, and in this case, gerbils, not just rats, as people often talk about when we talk about plague, and how those animal reservoirs interact. And so for the third plague pandemic, it was the lack of an animal reservoir in Europe, but also the activities of human populations, so surveillance and monitoring, and focus on hygiene and sanitation, which meant that populations could control the outbreak of plague and then end it. So if I've understood correctly, in order to understand plague, we actually need a variety of disciplines, because we need to understand all of these different arenas. And that's also the only way that we can understand the end of an epidemic. And it also sounds a bit like ways in which we talk about COVID-19. Is that correct? Nils, did you want to add?
I think the interdisciplinarity is a key here. Barbara's and my conclusion, together with colleagues, that plague must have come into Europe several times from outside. If you only look at the genetic information, the conclusion is that plague must have circulated around in Europe all along. But if you include ecological, historical, archaeological information, then that's no longer clear. Rather, the opposite, that plague must have come in several times. And we have to remember that although plague has ended in Europe, it is very active in many other parts of the world. In Central Asia, in Africa, in Americas, there are plague outbreaks all along. And the last one we had of a huge epidemic was in Madagascar a few years ago. So plague is a disease of today, but not in Europe, because we don't have the right condition for plague bacterium to circulate around or to live in a reservoir. But that is very much debated in the scientific field. I'm pretty sure we are onto the right answer.
It's always good to know that there's more research that can still be done and debates still to be had. So thank you very much, Barbara, and thank you very much, Nils, for sharing your expertise with us. And thank you all for watching our videos. Please do watch some of our other videos, and please also do fill out a feedback form. It only takes a few minutes of your time, and it helps to support research at the University of Oxford. Thank you very much.
Thank you.
Thank you.